Hello students, welcome on behalf of Science Academy. Today I am here with the second lecture of photosynthesis. In this lecture, we will discuss about the absorption and action spectrum, what happens to the light energy absorbed by the pigments, what is the photosynthetic unit, and concept of pigment system. Now what is absorption spectra? The degree of absorption of light by any particular pigment as the function of wavelength is referred to as the absorption spectrum for that particular pigment. Okay. Pigments absorb in different range of electromagnetic radiation, may it be visible or infrared. So different pigments will have different absorption spectra depending on their particular characteristics. Carotenoids absorb in the blue and green regions, while chlorophylls absorb in the blue and red regions. You can visualize the two different lines in the graph given in this slide of two different pigments which we have taken into account, chlorophyll A and chlorophyll B. We can see how much variety is there in the absorption of both the pigments at approx 500 nanometer, where chlorophyll B has maximum absorbance in comparison to chlorophyll A, and the vice versa is noted around 700 nanometer approximately, where chlorophyll A has maximum absorbance in comparison to chlorophyll B. This whole study of absorption spectra of chlorophyll A and B was done in ether. This diagram I have taken from my book. You can consider any diagram from internet. I prefer to study from the book, so I have taken it from the book. So now I have brought down the pigments, the important pigments which I have mentioned here, some of the important pigments and their range of absorption spectra. Like chlorophyll A ranges from 430 to 660, then chlorophyll B from 463 to 643, then bacteriochlorophyll A, B, C has particular ranges of their own. Please note it down. It may come in small questions or MCQs, and sometimes it's important in our question papers. Now what is action spectrum? Now we will study about the action spectrum of any particular pigment. Here we have taken in study chlorophyll A. The action spectrum is measured by plotting the response of oxygen evolution. What do you understand by oxygen evolution? The production of oxygen. Okay, it's nothing but the production of oxygen as the function of wavelength. The wavelength which is most effectively absorbed by chlorophyll A is the most effective in driving the photochemical reaction in case of chlorophyll A. Okay, so here we have taken it in the function of chlorophyll wavelength, and from this particular diagram you can see that the maximum absorption action spectrum is there around 700 nanometer. So that is their maximum range, and the ranges vary from 400 approx 400 to 700. So, if the pigments used to obtain absorption spectrum, say chlorophyll A here in the graph, are same as that cause the response, that is the action and the absorption spectrum match, which indicate that the light absorbed by the chlorophyll mediates oxygen production. Okay, so if the action and the absorption spectrums match, then it will indicate that the light absorbed by the chlorophyll will definitely mediate the oxygen evolution. Now let's move on to the next slide. Now what happens to the light energy absorbed by these pigments? Now you know what is the unit of light? We all know it's photon. So each photon represents a quantum of light. When light is absorbed, one molecule of chlorophyll becomes excited to a higher state. Then we all know that in the higher state or the excited state, the molecule is energetically very much unfavorable and it is not stable. So it has to return to its ground state, and this return to its ground state is possible in three possible mechanisms. In the next slide, I have inserted a diagram where I have shown that the possible three mechanisms by which light returns to its ground state. The first propaganda of the molecule would be to convert the extra energy into some other combinations of heat and light of a longer wavelength, and then return to ground state okay, and then return to ground state. So from the diagram it is very evident that The star mark represents the excited state of the molecule, and the normal molecule is represented without the star or the asterisk mark. While the second propaganda, what is it? If any chlorophyll molecule is available in nearby vicinity, it will transfer the energy into it. This process is known as resonance energy transfer. Now, what is resonance? You all have studied in chemistry, and I hope it will be clear to you. So, what is? I'll just give a short biological definition. Like resonance is something by which you share the electron pair. You share the possible energy by sharing the electron pair among the neighboring molecules, and that's what means resonance to me in simple biology. So, according to that resonance energy, the energy of the higher excited molecule, the electron pair, is transferred to the next molecule, and our chlorophyll molecule of interest comes to the ground state. This process is also known as Forster transfer. Okay. And the third process, in the third process, what happens? Any nearby molecule which can act as electron acceptor, which will accept electron, which is electron deficit, accepts the high energy electron and returns to the original state. Okay, and then it returns to the original state by taking a low energy electron from some other molecule which has excessive electron, that is, it is an electron donor. So I'll repeat whatever I said. Any nearby molecule which can act as electron acceptor accepts the high energy electron and returns to original state by taking a low energy electron from some other electron source, probably an electron donor. Now, possible mechanisms which are exploited in photosynthesis are mechanism number 2 and 3. Okay, keep this in note. Now, here in this slide I have written down whatever I taught you in the previous slide. So, please take a note of it. Okay, now we will study about the photosynthetic unit. So here is a small MCQ type question for you all, which is important, of course. So the question is, who were the first person to provide the evidence for involvement of chlorophyll molecule during photosynthesis? It was Sir Robert Emerson and William Arnold. I have forgot to mention the year, please note it down. They did this experiment in the year 1932. Okay, in the year 1932, Sir Robert Emerson and William Arnold. Now, what they did in the experiment? Now, they started the experiment on a certain suspension of alga chlorella. They performed the experiment on this. What they did? They subjected these suspensions to lights of saturating intensity for a very short duration. Might it be 5 to 10 microseconds, for a very short duration, okay? And then they determined the minimal amount of light needed to produce maximal oxygen during photosynthesis. Here maximal oxygen means they will record how much amount of light is required to produce 1 unit of oxygen during photosynthesis. That will basically work as a unit for a photosynthetic unit determination. Now, based on the number of chlorophyll molecules present in that particular suspension, they found out what? That 1 molecule of oxygen was released after absorbing 8 photons of light. Okay, so keep this in note. What happened? Based on the number of chlorophyll molecules present in that particular suspension of alga chlorella, what they found out? That 1 molecule of oxygen was produced after absorbing 8 photons of light. And how many chlorophyll molecules were present in that particular suspension? It was 2400 chlorophyll molecules. So they came to a conclusion that approximately 2400 chlorophyll molecules from that suspension acted as a single unit for producing 1 molecule of oxygen against 8 photons of light. So this 2400 chlorophyll molecules acted singly in a certain unit and constituted as a certain unit. So this unit was known as photosynthetic unit for that particular suspension. Now this varies from any photosynthetic organism to any other photosynthetic organism. The size of photosynthetic units vary as mentioned in the slide. It ranges from 300 to 5000 chlorophyll molecules depending on our species or the photosynthetic organism we have taken in account. Now lastly we will study about the concept of pigment system. How did the concept or how did this pigment system came into work, or who discovered it? Now it was Sir Robert Emerson and his colleagues again in the year 1943. What they did? Sir Robert Emerson and Clarton Lewis examined the action spectrum of oxygen in green algae. Which green algae they used? Chlorella pyrenoidosa. Please keep in note, okay? And to their surprise, what they found? That the quantum yield was fairly constant up to 680 nanometer, after which it declined. That means in the far red zone it declined, but before that it was fairly constant. Everything is working properly. So first of all you should take this into account. What is quantum yield? It is nothing but the number of oxygen molecules produced per photon absorbed. So it has a direct relation to the action spectrum. The purpose of this experiment suggested that wavelengths shorter than 680 nanometer are very much efficient in the process of photosynthesis. They withdraw this conclusion from this particular experiment. Later, in some other year, they did another experiment with two set of beams. Okay, with two set of beams, in which what they did, one was in far red region, that is the wavelength was greater than 680 nanometer, and the other was in red region, that is wavelength was equal to or lesser than 680 nanometer. And then they found out that the constitutive action of the two beams increased the rate of photosynthesis 2 to 3 times than the action of any single beam. So can you imagine? The effect was not only doubled, it was tripled on the constitutive action of both the beams. Whereas in each single beam effect we will not getting that optimum photosynthetic activity output which was required in the case of which was required for the plant to photosynthesis. Like experimental results and the practical results were not same, so they did this experiment to show that the constitutive result was something different, and the separate results of both the separate results of both the beam of light was totally different. So what they concluded from this, that the enhancement effect provided special evidence that photosynthesis involves two photosystems, one driven by light of long wavelength which is 680 nanometer and the other by short wavelength which is lesser than 680 nanometer. This whole experiment is termed as Emerson enhancement effect. What? Emerson enhancement effect. Now this is the picture of Emerson enhancement effect. It is very clear. This is also taken from a book. Okay, you will get many colorful pictures in internet if you want. Now see, you can see it from the picture very clearly that on switching on the two beams distinctively there was low rate of photosynthesis, but when both the lights were summed up the effect was much more, or actually triple times the effect which was seen on switching on the two beams distinctively. So the constitutive effect was thrice more than the previous effect. So overall this effect was known as enhancement effect. And what was concluded from it? That photosynthesis involves two photosystems, one driven by light of long wavelength which was 680 nanometer, greater than 680 nanometer, and the other of short wavelength which was lesser than 680 nanometer. Okay, so this was all for today's lecture. Okay, so this was all for today's lecture. If you find this information useful, please subscribe to our channel, to hit the like button if you find the information useful, and please subscribe to our channel and hit the bell button for further notifications if you want to remain updated with our video contents. That's all for today. Thank you, and I will be uploading more videos on the lectures of photosynthesis further, so please stay tuned. Thank you.